when is this any gene or allele which is present at the recessive condition? That time recessive means recessive homozygous more specifically. That time it will be showing the harmful effect. But now what happen if you produce a hybrid variety? So that harmfulness effect they get suppressed. So that suppressedness of the harmful effect, simply these are called as the hybrid vigor. So we are talking about some information. What is meaning of that hybrid vigor? Hybrid vigor nothing but, is this any homozygous recessive allele? We are talking about allele, okay? Means alternative form of gene. That is recessive homozygous. You know very well what is meaning of homozygous, huh? So recessive homozygous allele present. If recessive homozygous allele present, that time they are showing some harmful effect. So might be possible these are the showing some harmful effect. Harmful effect. But next step. But if hybrid condition is present, you know very well what is meaning of this hybrid. One is recessive, another one is dominant, right? So if hybrid condition is present, if hybrid condition is present, that time that harmful effect gets suppressed. Then that particular harmful effect, it's get suppressed. Okay, so these are the simple process. These are called as the hybrid vigor or simply called as the heterosis. So to explain the heterosis, I'm taking one example so you will clearly understand what I want to say. So should I continue with example? Because see, all these are the simple concept, and if you understand the example, ultimately you can remember what kind of this, how to create a definition. Yeah, should I continue? Should I continue with example? Okay, Sahil. Chalo. So now to explain the hybrid vigor, simply we are taking one example, that is cross between the two parents. Now ये parent A है, and now these are the parent B. Parent A, and now these are the parent B. Now parent A, it has the genotype capital A, capital A, capital B, small c, small c, small d, and small d. 4 genes are present, and out of that 4, 2 dominant, 2 recessive. Now, in another parent B, there is a presence of genotype small a, small b, capital C, capital D, capital D. Same scenario, but now what happens? C and D, these are the present in dominant stage. Okay, C and D, both these are the in dominant. So we have to represent how they represent, means what kind of this gene represent for the which condition. So if we are considering dominant gene together, this dominant gene responsible for high protein content, and this high protein simply represented by, these are the product of the two gene, A gene and the B gene, because A and B they are present in dominant condition. But now what happened? Due to that recessive character, homozygous recessive, see, small c recessive, both these are the homozygous, right? So due to that homozygous condition, now they are harmful, showing the harmful effect. And which kind of this harmful effect? That is sensitive to disease. These are sensitive to disease. And these are the character due to small c, small d. Okay. So these are the scenario with respect to parent A. Now we have to discuss with the parent B also. In case of this parent B, again there is a presence of these. How many genes? There is total 4 genes. Out of that 4 gene, पहले जो 2 genes है, small a, small b. So definitely these are the 2 genes which is code for the protein. But now what happened in this condition? there is a production of the less protein because gene are present in recessive condition. ठीक है, तो यहाँ पे loss हुआ. These are the harmful effect because these are the present in recessive. But now what happened? Due to there is a presence of this capital C, capital C, capital D, capital D. Now these are showing resistance to disease. So improve variety, improve quality, resistance to a particular disease. So now what happened? What we understand from this point? If you consider recessive homozygous condition, दोनों parent में consider करो, right? यहाँ पे consider करो, that is small c, small c, small d, small d. Yes, these are showing the harmful effect. And in case of small a, small a, small b, small b, recessive homozygous condition, and still these are showing the harmful effect. So now what happened? We want to reduce, we want to suppress that harmful effect. तो हमें क्या करेंगे यहाँ पे? We have to perform the hybrid vigor. Hybrid vigor means what? Cross between the parent A and the parent B and produce a superior quality hybrid variety who having high protein amount as well as having resistance to disease. So now what happened? Just simply we are performing the hybridization process between them. Cross करो parent A, parent B, and now what we obtain? We obtain a superior quality of the hybrid variety, and these hybrid variety having the genotype, that is capital A, small a, capital B, small b, capital C, small c, capital D, and the small d, right? And now what happen? If you see what kind of this phenotype they produce, so definitely whatever that protein amount is present in that particular hybrid plant, that protein amount is high, and such type of this plant, these are showing, these are the resistance to disease also, resistance to disease also. So now what happened? Here we produce a hybrid variety, and what is meaning of this hybrid variety? We want a produce a superior quality. But now what happened? Why we required this thing? Because when a recessive allele present in homozygous condition, they produce the harmful effect, right? But now what happened? These hybrid, whatever they produce, these hybrid can suppress the harmful effect. Yes, definitely they can suppress the harmful effect, right? So such type of this phenomena, production of this hybrid. But what is the aim? What is the aim to produce the hybrid? We want to reduce the harmful effect. We want to suppress the harmful effect because we know that according to Mendelian dominance recessive, अगर single dominant gene भी present होगा, single dominant, so definitely the dominant gene get expressed and the recessive character it can mask itself, right? So यहाँ पे भी same scenario है. Now capital A, capital B, capital C, capital D. Single dominant gene are present, but you know very well dominant gene which is always and always expressed according to Mendelian inheritance, right? So yes, we produce the hybrid, but now this technique, these are called as a hybrid vigor, also called as a heterosis, because we want to suppress recessive homozygous character because these are showing the some harmful effect. Got it? This point is clear, everyone? Just quickly check the entire structure. Very simple concept है, ज़्यादा कुछ rocket science नहीं है यहाँ पे. Very simple, straightforward. Simply we are talking about harmful effect. That is, we want to suppress a harmful effect which is created due to homozygous recessive. Okay? Yeah. चलो continue करो. Once you done, please tell me, huh? Now next point we are talking about inbreeding depression. I hope आपको ये पता होगा because it's similar to evolution depression. Okay, chalo, done. So next point we are talking about inbreeding depression. Write down the title. थोड़े छोटे छोटे concepts है. First of all, we cover that point. So simply we are talking about inbreeding depression. Very simple. Now as it is we are taking the hybrid variety. तो हमने यहाँ पे produce किया capital A small a, capital B small b, capital C small c, capital D small d. Now these are the one species, that is the hybrid species, which is produced by heterosis process, which is produced by the hybrid vigor, right? Hybrid vigor है ये, because we want to suppress the harmful effect. So yes, whatever we produce the hybrid variety, these are having the high protein amount as well as these are showing resistance activity against the disease. But now what happened? Now we are doing continuously self pollination. Suppose ये hybrid variety है, right? And this hybrid variety we are carried out continuous self pollination again and again. बार बार हमें self pollination करना है. So now what happened? Due to self pollination, various time continuously self pollination process means what happened? This hybrid variety should be crossed with the hybrid, right? Selfing. Self pollination means we are talking about that is selfing. So now what happened? Due to that selfing, एक बार ऐसा stage आएगा at the final stage where homozygosity will increase. At the end, these are the responsible for homozygous condition, it will be get increased. Now what happened? Homozygous condition means what? Might be possible recessive allele they again reappear. Might be possible. Huh? Now what happened? Homozygous condition is increased. It means that we are talking about that recessive allele जो पहले suppress हुआ था. Recessive allele. Recessive allele means just we discussed there is a small a or small b or might be possible small d or the small c any character. So we are talking about recessive allele which is present in homozygous. यहाँ पे लिख सकते हो homozygous recessive allele. So that homozygous recessive allele they can reappear. Disappear हो गया था due to hybrid vigor. But now what happened? If you are continuously performing selfing, that is self-pollination. So due to what happened? Homozygosity increase होगा, right? If homozygosity is increased, ultimately what happened? That homozygous recessive allele जो पहले hide हुआ था, might be possible they can arise. Can be arise. And once such homozygous recessive allele arise, it means that quality of that hybrid they decrease. Hybrid vigor which is be produced, right? So that quality of that hybrid ultimately it will decrease. So finally what happened? That is harmful effect arise, and this harmful effect nothing but we are talking about that is hybrid vigor loss hoga. Hybrid vigor or hybrid quality. First of all we are talking about quality. So hybrid quality it will be decreased, or in other language you can say that is vigor. Vigor means hybrid, दोनों का मतलब same ही है. So we are talking about that is hybrid vigor qualities get decreased. Okay, and now what happened? Once हमने इतना मेहनत लिया, we produce parent A cross with the parent B, and now what happened? Finally we what we obtain? We obtain loss of that hybrid, loss of that quality. So such process simply these are called as inbreeding depression. Continue performing the selfing, and if continuously selfing self pollination, so ultimately what happen? Superior character it will be get loss. So that's why sometimes कहा जाता है blood relation में शादी नहीं करनी चाहिए. Normal thing है, right? During the blood relation marriages get harmful because now what happen? In blood relation basically same kind of this means genotype का ज्यादा variation नहीं होता, no variation in allele. So now what happen? Might it be possible कि कोई disease appear होता है, या फिर sometime what happen कि कोई new quality नहीं होता. New quality of gene means what? For example, if your family is having some whitish color, for example skin color, पूरे के पूरे person whitish color के, right? So और relatives में शादी कर ली तो ultimately whatever the progeny produce that is having the same body color, right? कॉम्बिनेशन कोई नहीं होगा. वही आप अगर कोई फेयर से या फिर किसी ब्लैक से शादी कर रहे हो, सो अल्टीमेटली व्हाट हैपन? इट विल बी क्रिएट्स अ वेरिएशन. फॉर एग्जांपल सिंपल बोला हमने, राइट? सो That's why when you are doing this continuously selfing, so ultimately whatever the superior qualities are present, the superior qualities it get decrease. Hybrid loss, hybrid vigor quality decrease, right? So that concept simply it is called as the inbreeding depression. Got it? This point is clear, everyone? Done? Okay, Nathan. Okay, chalo. So this part is over. We discussed the two concepts. Number one, hybrid vigor. Number second, that is inbreeding depression. Now, next point we are talking about थोड़ा सा large है, methods of crop improvement. Write down the title. We'll discuss different methods of crop improvement. In our syllabus, total 4 methods are given. First one, that is hybridization. We just discussed by taking so many examples, parent A cross with the parent B and produce the superior quality of the plant. That's it, hybridization. So we complete this part. Second point, that is polyploidy. We discuss this part later. Third one, that is mutational breeding. Create a purposefully mutation to produce the superior quality of the plant. Mutational breeding. And the next one, that is selection. तो हमें selection भी करना पड़ेगा after mutation, right? So under the crop improvement program, these are the total 4 methods. We just cover the hybridization. जो लिखना है आप लिख सकते हो. Take any example. You can add any property. Parent A, parent B, name, genotype, properties, cross करो, hybrid करो. That's it. Simply these are called as a hybridization. Okay. And if you want, if you are taking the bisexual flower, that time you can explain the emasculation, bagging, debagging, dusting, now rebagging, and the labeling. So you can explain that point also. Okay. Chalo. Yeah. So we continue with the first point. Hybridization हमने cover किया है. So now we are talking about second type with respect to crop improvement method, that is polyploidy. Second method we are talking about, that is polyploidy. Polyploidy means what? More than 2 sets of chromosomes are present. Poly means many, right? And normally what happen? Whenever we are talking about organisms, like specifically we are talking about plant diploid hota hai. 2 end, right? Diploid condition. So number of the chromosomes get vary, but now in this condition we are talking about more than 2 sets of chromosomes are present. So that condition is represented as a polyploidy. So polyploidy, nothing but simply we are talking about, that is more than, don't worry, we'll take the example for the explanation. More than 2 sets. More than 2 sets of chromosomes present. Polyploidy basically is divided into 2 types, atopolyploidy and the allopolyploidy. So we continue this part. Polyploidy divided into 2 types. First one, atopolyploidy. And the next one, that is allo. That is different. Allopolyploidy. When we are talking about atto, it means that same set of chromosome get multiplied. Basic and the same. Atto का मतलब same ही होता है, right? So what is meaning of this atopolyploidy? It means that is same set of chromosome, same set of chromosome, it is get multiplied. Okay, so such type of this polyploidy simply it is called as एटोपोलिप्लॉइडी। सो मान लो कि यहाँ पे कोई प्रेजेंट है, सेम सेट मीन्स कैपिटल ए, सेम सेट ऑफ क्रोमोसोम, राइट? नाउ इट इज गेट डबल, now it is get multiply, so now it is get what they form, capital A capital A capital A capital A, four times, two set थे, now इसका means actually one pair capital A, now they form the another pair, so multiply, same set of chromosome. And what is meaning of this allopolyploidy? Simply what happened, there is a presence of capital A capital B, अब यहाँ का multiplication होगा, means they produce capital A capital A capital B capital B. So what is meaning of this allo? Allo means different set of chromosome get multiplied. Different set of chromosome they get multiplied. Okay, that's it. Very simple. Atopolyploidy and the allopolyploidy. Chalo. Now, in case of polyploidy, we are taking few examples. We are not taking too much detail. कैसे क्रॉस बना, क्यों बना, what is the number of the species, we talk about the start point and the finally end point, middle का सब कुछ हम skip कर रहे हैं because not important. So now we are taking some polyploidy methods, natural as well as the artificial, which is included in our syllabus. So first of all we start with the natural. Should I continue further part with polyploidy? Okay, Deepika. Yeah. So next part we are talking about, write down. To explain this polyploidy, simple we are taking the example condition with respect to natural polyploidy. So first of all we are talking about example natural polyploidy. And in case of this natural polyploidy we are taking the wheat variety. So there is a one question, very common question. Origin of bread wheat. Sorry. Origin of the bread wheat. The wheat which is used for the bread preparation. So name of the that species, wheat species, that is Triticum aestivum. Triticum, species name aestivum. So we are talking about how that Triticum aestivum species it is get originated, and these are the bread variety which is used. Brown bread आपने नाम सुना होगा, right? So nothing but we are talking about that is wheat which is used for the bread preparation. This wheat example of the natural polyploidy. Natural mutation हुआ, right? And because of this natural polyploidy means there is a change, and one species it is and now this another it is converted to, and finally we obtain the one species that is Triticum aestivum. So we are talking about how that Triticum aestivum species is get originated, and that Triticum aestivum common name also called as the bread wheat. Should I continue? Should I continue? Okay, fine. Chalo. So very more complicated process actually, so we are not taking too much complication. I'm taking very simple scenario because here we want to produce one hybrid stage. Hybrid pehle sterile hoga, now natural duplication process, and now they get double, become they fertile, that's it. We are not taking too much complications. So now in this condition, first of all we are talking about there is a one species of the triticum, that T for the triticum. First of all I write down. So T for the triticum, genus name and species name Boticum. Triticum Boticum. These are the first species. These first species basically in diploid condition it has total 14 chromosomes. So I'm writing the ploidy level also. At diploid condition there is a presence of this 14 chromosomes. Okay, कितने chromosome है 14. And genotype which is present, this genotype it has capital A capital A, only one type of gene, capital A capital A. So this capital A, nothing but simply we can say it has the total 14 chromosome at the diploid condition. Now what happened? This triticum boticum undergo natural mutation. Natural mutation समझ में आता है? Natural means suddenly ऐसे ही कुछ तो हो गया. Natural mutation. So these are the undergo the process of the natural mutation. Slow होता है natural mutation, but it is happening. So accidentally it undergo the natural mutation. And after this natural mutation, now it is converted into triticum monococcum. So it is converted into T for the triticum. I'm not writing the triticum entire. And now it is converted into monococcum. It will be form a species. If you are talking about what is diploidy level, यहाँ पे भी same था. That is diploid condition AAA, and number of the chromosomes, these are the same, that is total 14 chromosomes. Now what happened? Eventually, naturally, this triticum monococcum cross with the some grass species. Naturally हो गई. These are the not in, because see, wheat also monocot as well as the grass also monocot. So it naturally हो गया. Accidentally it is happening. So now what happened? This triticum monococcum naturally cross with, naturally hybridize with the grass species. So I'm giving the sign for the cross. Cross means what? Hybridization. Remember this point. Natural सब कुछ हो गया, कोई induce नहीं है, because we are talking about natural polyploidy. So now naturally that monococcum species, it is crossed with the some grass species, and more specifically we are talking about goat grass. Goat grass species naturally cross हो गया. And now what happened? The genotype which is present, that is 2N, capital B, and the number of the chromosome, these are the total 40. Just remember this, what is the condition is happening. Triticum, boticum, these are the wild species. ये wild species था. Initially ये present था. These are the wild wheat variety which is present. This wild wheat variety undergo the natural mutation. After the natural mutation, it is converted into monococcum. कुछ तो difference हुआ होगा. We are not taking because there is a variation with respect to grain. पहले grain की size means that what we say, inflorescence height is larger in case of this wild species. After the natural mutation, size of this inflorescence, it is get reduced. Monococcum, size of inflorescence, wheat inflorescence, that size it is get reduced. That is the effect of mutation. So none of our syllabus, so we are not taking that part. So now what happened? This wheat variety who having capital A at diploid condition, total 14 chromosome, crossed with the goat grass species, because see, wheat भी monocot है, grass भी monocot है, chromosome में 14. But now what happened? Because of such cross, यहाँ पे हो गया hybridization. Between these, there is a process can be occur, that is hybridization. Because two different species, they showing hybridization, they fertilize with each other. And now what happened? After the hybridization, हमें obtain हुआ एक hybrid variety. We obtain a hybrid variety, and please remember, ये जो hybrid variety है, these are the sterile hybrid variety. These are the sterile hybrid variety. Why sterile? Definitely at diploid condition, capital A gene, capital B gene, and the number of the chromosome, these are the 14. But if you see, why we say these are the sterile one? Because there is a presence of single capital A and the single capital B. It means that homologous chromosomes are absent. These are the non-homologous chromosome. Yes or no? हम homologous किसको कहते हैं? Capital A, capital B, because A pairs with A, B pairs with the B, right? Chromosome number 1 pair with the chromosome number 1, chromosome number 20 pair with the chromosome number 20. That is called as the homologous condition, right? But now what happened? There is a presence of gene, that is capital A, capital B. But in this scenario, these are the non-homologous chromosome. And when we are using non-homologous, it means that homologous partner is absent, right? So these are the non-homologous. These are the non-homologous chromosome. And when we are talking about non-homologous chromosome, it means that these are sterile variety. Because see, अगर homologous partner absent है, it means that there is a no crossing over, no process, further process. So such type of this species, these are the totally sterile. Now what happened? These sterile species naturally undergo duplication, polyploidy. Naturally, सब कुछ naturally हो रहा है, कुछ भी यहाँ पे नहीं हो रहा है. Okay, these are the naturally everything is happening. So now what happened? Next step, this sterile hybrid variety. This sterile hybrid variety, now these are naturally undergo duplication. Duplication of chromosome. So now tell me what kind of the genotype we produce? Tell me. Sterile hybrid tha, it has the diploid condition AB is equal to 14. Now tell me, if they undergo the natural duplication process, now what kind of genotype they produce? Think. Double करना है हमें. Duplication of chromosome. Shantanu, very good. Very good. And 2n is equal to total 28. Very nice. चलो अब समझ में आ रहा है आपको. So now what happened due to natural duplication of chromosome? Now there is a formation of fertile hybrid. पहले sterile था. Now it is converted into fertile. And now why we say fertile? 2n is equal to capital A, capital A, capital B because homologous chromosomes are absent. And now 14 का double हो गया. Means how many chromosome? Total 28 chromosome. How many chromosome? Total 28 chromosome. Got it? This point is clear, everyone? Now what happened? That chromosome number it is get double. So when you are using such condition, why we say that is fertile? Because now in this condition, these are homologous chromosome. So, these are the homologous chromosome. So homologous chromosome means partners are present. So definitely what happened? We produce one the fertile species. Got it? Yeah. So now what happened? These fertile species which is get produced. Now we have to give a name because these are the fertile species. पहले कैसे था? पहले these are the having only 2 set of chromosome. Now it is become 4 set of chromosome. So now it is become tetraploidy, right? So this whatever we produce, this fertile hybrid, nothing but we are talking about these are the tetraploidy. Tetra, 4, right? Tetraploidy. And why we say that is tetraploidy? Because at diploid condition there is a presence of capital A, capital B is equal to total 28 chromosome. So tetraploidy ban gaya. 4 set of chromosome. Tetraploidy, right? And now the name of this tetraploidy, that is Triticum turgidum. So I'm giving the name. इतने सारे नाम याद रखने की जरूरत नहीं है. Just remember the thing. So we are talking about this name. चलो नेम नहीं लिखते, डायरेक्टली हम म्यूटेशन के बारे में बात करते हैं. So this tetraploidy, whatever this produce, this tetraploidy again second time it is undergo the natural mutation. Again it is undergo the process of the natural mutation. See, homologous partner present है, tetraploid form हुआ. Now they form the fertile hybrid, right? But now what happened? Eventually they undergo the natural mutation. And after the natural mutation, there is a formation of a very, very specific species of the wheat variety, that is Triticum durum. D-U-R-U-M. T for the Triticum and durum. D-U-R-U-M. These Triticum durum species, these are the used for the preparation of the pasta, maida, noodles as well as the macaroni. So these are having some economic importance. It is used to preparation of pasta, maida, noodles and as well as macaroni. Okay, so that's why this Triticum durum species also called as the मैक्रोनी wheat because ये थोड़ा हार्ड रहता है. So these triticum durum also called as macaroni wheat, which is quite hard. It has hard in nature. Got it? So just check the steps. Steps important क्या है आपको याद रखना है, which is the wildlife species name. Now you have to remember that is Triticum durum, which is used for the preparation of the pasta maida, noodles and the macaroni because of its hard nature. Got it? Got it? Okay, chalo. So now we are talking about these are the one condition when that hybrid fertile hybrid undergo the natural mutation. तो ये हुआ. Now I am talking about second scenario. Same this fertile hybrid variety that is having 2n is equal to capital A, capital A, capital B, total 28 chromosome. Now they undergo the again hybridization, natural hybridization, and finally they produce the triticum estivum. So chalo, we are talking about the next point. It's the first point. Second point, same we are talking about that condition, that is capital A, capital A, capital B, capital B. ये क्या था? These are the hybrid variety which is be produced at the last. Hybrid and 2n is equal to capital A, capital A, capital B, and the total number of the chromosome. These are the total 28 chromosome. Same variety हम ले रहे हैं, okay? Because same that hybrid variety form the macaroni wheat, and now we are talking about second changes. This hybrid variety now it is undergo natural hybridization. एक हमने देखा mutation. Once it is undergo the mutation, they form the macaroni wheat. Now we are talking about same that hybrid variety. Now it is undergo the natural hybridization. Natural hybridization. And after the natural hybridization, now they form a variety. That variety nothing but we are talking about capital A, capital B, capital R, which is the sterile. Sterile one. Now this sterile it is become fertile. कैसे बनेगा? Natural undergo the process, that is formation of this capital B, capital A, capital A, capital A, capital B, and the capital R, and finally it is become fertile species. And how these are the possible? It is possible because of the duplication of the chromosome, natural duplication. You are not using any chemical. ये naturally हो गया. Sterile species it is converted into fertile, right? And now what happened? These fertile species which is get produced, these has ploidy level, that is 2n is equal to total 42 chromosome. And now this species nothing but these are called as the bred wheat, final species bred wheat, and this bred wheat simply these are called as Triticum estivum. Triticum estivum. And if you see what is the ploidy level of this Triticum estivum, so these are the hexaploidy. Why we say that is hexaploidy? Because total 6 set of chromosomes are present. A, B, B, R. Simple meaning है, no need to remember the middle step, कैसे form हो रहा है, क्यों form हो रहा है. Just remember the first and the last point. So you have to remember only this point, that is this point. That is Triticum estivum. These are the hexaploidy, and hexaploidy means what? We are talking about there is a presence of the total 6 chromosomes, and the 2n is equal to total 42 chromosomes are there, which is present in 6 set. And that Triticum estivum, common name also called as the bred variety, or sorry, bred wheat, which is the hexaploidy. Quickly check this scenario. थोड़ा सा complicated है, but no need to remember each and every step. Just remember the first step. Now next step, that is macaroni कैसे form हुआ, and finally step remember the last one, how that Triticum estivum it is get formed. Because यहाँ पे simply process हो रहा है. Which process? duplication, natural hybridization, natural mutation. 3 steps it is happening. Done this part? Just quickly check. Triploidy. 3N means triploidy. Shantanu, triploidy means ABC. These are the ABR. These are the triploidy, but now these are the sterile because homologous partners are absent. These are the sterile one. Okay, chalo. It's not represent as a 3N because see homologous partner is what? One it is coming from the mother, another one is coming from the father. So that's why we are talking about in most of the plant, except few conditions like banana. Banana it is a natural example of the triploid. So except banana, maximum whenever we are talking about plant species, these are the diploid in nature. So in case of the diploid, kitne bhi number of the set chromosome ho sakte hain. So here we are talking about 6 chromosomes. So that's why we are talking about these are the hexaploid. Okay, chalo. Should I continue next one? Artificial polyploidy. Okay, chalo. So there is a one question from the artificial polyploidy. Again, we are taking one variety that is triticale, which is the variety means what we say. These are the combination of the rye and wheat variety. So after the natural, we are talking about second step, artificial polyploidy. Artificial polyploidy. And in this point, we are talking about one example that is triticale. And please remember triticale, basically these are the first man-made crop, hybrid crop. These are the first man-made crop, wheat variety, that is triticale. So we are talking about triticale, basically it has the two types, octaploidy as well as the hexaploidy. So it has the two types. Number one, there is a presence of this hexaploidy, and the another one that is octa, 8. Octaploidy. So first of all, we are talking about that is hexaploidy. In case of this hexaploidy, basically scientists selected a wheat variety, Triticum estivum, and the another wheat variety that is Triticum turgidum. And these two species cross with the rye, that is Sickle cereals. So simply we are taking simple explanation. So first of all, we are talking about wheat variety, T for the Triticum, and the species name that is turgidum. So these are the wheat variety. This wheat variety, it has 2n is equal to capital A, capital A, capital B, and total number of the chromosome that is 28 at diploid condition. Now this Triticum wheat species, wheat variety, they cross with the rye, and the scientific name of the rye that is Sickle. Sickle cereal. And the genotype that is diploid condition, presence of capital R, that is 40. These are the genotype. So if you want, you can write down simple meaning. These are the wheat variety, and now these are the rye, common name, wheat and the rye, because triticale, these are the combination of wheat and the rye. No need to remember the scientific name. Again, I'm telling you, no need to remember. Now, so these two species undergo the process of hybridization, but now it's not a natural hybridization. These are the artificial hybridization. Artificial hybridization. And after the artificial hybridization, now what happened? There is a production of sterile hybrid variety. So now there is a formation of this sterile hybrid variety. Definitely sterile hoga because homologous partner, these are the absent. And whenever we are talking about, these are the, what we say, that is ploidy level, that is 2n is equal to A, B, R, and we are talking about chromosome number total 21. Yes, यहाँ से आ गया 14 plus 7 21. Yes, true. So these are the 21, which is the sterile one. Now what happened in this scenario? This sterile hybrid variety, it is undergo the duplication, but now यहाँ पे use किया one drug, that is colchicine. Which drug used? Colchicine. One question from the colchicine. Only one question, right? No need to remember genotype, whatever, सब कुछ भूल जाओ. Colchicine. And what is the colchicine? Colchicine basically it is induced duplication of chromosome, artificial duplication. Okay, it is induced duplication of chromosome because during the last class means last year in cell cycle cell division हमने पहले discuss किया था. Colchicine basically block the metaphase. Block the means it is attached, it's bind to the spindle fiber. And what is the function of the spindle fiber? Spindle fiber responsible for the pulling of the chromatid during the karyokinesis. So now what happened? Replication to hoga definitely, but there is a no nuclear division. So now what happened? Inside the one cell, there is a presence of the many chromosome. Duplication of chromosome because colchicine bind the spindle tubules and they inhibit the karyokinesis. They inhibit the cell division. So replication to ho raha hai in S phase, but now there is a no division. So now colchicine drug play a very important role. So one question from the colchicine, induce the duplication process. Chromosome number ko double karega. So now what happened due to colchicine treatment? Now that sterile process, sterile hybrid, it is converted into fertile. It is converted into fertile hybrid. So chromosome number क्या हो गया? Double हो गया, right? So now in this condition, there is a formation of 2n is equal to capital A, capital A, capital B, capital B, capital R, and 21 का double means it is converted into 42. So now ये जो variety बना, finally, these are the fertile offspring, and name of these are fertile offspring, that is triticale. But that triticale, these are which ploidy level? Hexaploidy. Okay, चलो एक मिनट. I think this screen is frozen. So finally what we obtain? We obtain this variety, fertile hybrid variety, and this variety nothing but we can say these are the hexaploidy triticale variety, which is a combination of wheat and the rye. Okay, that's it. Last point पे question. First step and the last step and colchicine. That's it. बीच वाला सब कुछ भूल जाओ. Yeah. Same scenario with respect to octaploidy. Should I continue? Okay. So same condition with respect to octaploidy. यहाँ पे भी same condition होगा. But now in this condition we are taking this species that is Triticum aestivum. Triticum aestivum. Now they cross with the rye. Sickle. Cereals. Now these are the condition with ploidy level. 2n is equal to capital A, capital A, capital B, capital B, capital D, capital D. And chromosome number, these are the 42. with respect to triticum aestivum. And finally, with respect to rye, there is a presence of diploid condition, presence of capital R, that is total 14. Same hybridization process hoga, sterile progeny get formed, now the colchicine treatment, and finally we obtain octoploidy triticle. Everything will be same. Hybridization, formation of the sterile hybrid. Okay. Now the sterile hybrid undergo the process of colchicine treatment. After the colchicine treatment, we obtain the fertile variety. And these are the fertile hybrid, nothing but we are talking about, that is 2n is equal to capital A, capital A, capital B, capital B, capital D, capital D, capital R, octaploidy बन गया. And the total number of the chromosome, now these are the 56. And the name of this species which is get formed, these are the tritical, but now more specific we are talking about octaploidy tritical. Octaploidy tritical. So last का आपको याद रखना है, that's it. So question it is coming from the tritical के कितने varieties है? 2 varieties. One variety that is hexaploidy, another variety that is octaploidy. Hexaploidy having the total 42 chromosomes and the octa having the 56 chromosome. And the another point, these tritical, these are the combination. These are the what we say hybrid. These are the first man-made crop and fusion between the wheat and the rye. Got it? This point is clear? Okay. So question वाला part देखते हैं. Why we produce such type of this hexa and the octaploidy of the tritical? What is the benefits of tritical? So we are talking about last point with respect to tritical. What is the important points? Why we produce this tritical? Man-made है, colchicine use किया. What is the requirement? So now what happened? These tritical species, we are talking about both the hexa as well as the octa, common है सबके लिए. So this tritical basically it is having the high yield capacity. And this high yield capacity due to wheat. Because it is a combination of wheat and the rye. So wheat having the high protein capacity, so ultimately yield iska bhi zyada hoga. Now what happened? In case of rye, there is a very good property with respect to rye, that is rye, it has the disease resistance activity. So these are the resistance to disease. Whatever the new species it is get formed, these are the resistance to disease. And why these are the resistance to disease? Due to rye. Because inside the rye, there is a specific properties are present, and due to that property, now that hybrid property which get formed, hybrid plant, that is octa as well as the hexaploidy, they showing the resistance activity. Number third, it having the high protein amount, but they having low gluten. Gluten, these are the protein which is present inside the wheat, right? But इसके अंदर क्या present है? These are the consist of the low gluten. So because of low gluten, such type of this critical variety used as a fodder crop. These are the used as a fodder crop. So you know fodder crop means what? That is animal feed plant, right? So definitely it has the high protein amount, but gluten low hai, right? And due to that, because of this property, such type of this hexaploidy tritical as well as the octaploidy tritical, these are the artificial polyploidy plant which is man-made plant by using the colchicine drug, such type of this plant, they are used as a fodder crop plant. Got it? Okay, Abida, this point is clear. What is the important point? Remember this point, okay? Kitna genotype hai, diploid condition, what? No, not necessary. It's for your understanding purpose ke how it is going on. Okay? So just solve the previous year question or read the NCERT, you will get the clear-cut idea कि आपको किस limitation तक पढ़ना है. So colchicine important है, and now this point is important. What is the uses of that critical? Got it? Got it? Kinmin, Tanisha, Risa, Nelia, Abida, Sahil, Shweta, Malaika, Hiba, Nathan. Okay, very good. Chalo. Now all these are the information about the second point of the crop improvement method, means crop method, methods of the crop improvement. That is, we discussed the hybridization. Now we complete this part, that is polyploidy. Next point we are talking about, that is mutation breeding. Should I continue? Okay, Kavya. Yeah. So third step, mutational breeding. Mutational breeding. Very easy, very simple question. Mutational breeding means what? Mutation means what? Something a sudden hereditary change in genetic material of the organism. So now what happened? Mutation दो types के होते हैं. Spontaneous mutation also there, as well as the induced mutation also there, right? And whenever we are talking about the mutation breeding, we are talking about induced mutation. We are not taking the spontaneous mutation, natural mutation. Okay. I hope in evolution you already discussed the mutation type, spontaneous and the artificial, natural. So I'm here talking about only one point, that is induced mutation. Purposefully we have to create a mutation. Now whenever we are talking about that is induced mutation, first method, use of chemical mutagen. Chemical mutagen used. Mutagen means what? Who create the mutation. That is called as the mutagen. Chemical means what? Certain kind of this chemical. So this chemical mutagen, nothing but simple, we are taking the example, that is mustard gas. So mustard gas basically it is used to create a mutation, purposefully mutation. Another mutation agent, that is physical mutagen, that is physical agent who create a mutation. And in case of this physical mutagen, mostly two rays are used. Number one, X-ray. Generally, X-ray use किया जाता है, and this X-ray basically used in Drosophila. In case of animal, mostly X-ray bombarding technique, it is used to create a purposeful mutation. But whenever we are talking about plant, in case of this plant, gamma rays mostly used. In case of this plant, X-ray used for the animal, and the gamma rays mostly it is used for the plant to produce the, what we say, that is mutation, purposefully mutation. Now what happened? Whenever we are talking about this mutational breeding, definitely pioneer of that plant mutation breeding, whatever the work it is, goes to Dr. M.S. Swaminathan. And we know Dr. M.S. Swaminathan, he was the father of the Indian Green Revolution. So we are talking about, that is one scientist name, that is Dr. M.S. Swaminathan. One question from this point, okay? Dr. M.S. Swaminathan. Dr. M.S. Swaminathan, basically, he was the pioneer, means first man, pioneer for plant mutation breeding program. Plant mutation breeding program. इसने पहले साइंटिस्ट ने, स्वामीनाथन ने पहले चेक किया था, yes it is possible or not. As well as basically when we are talking about, that is, he is the father of Indian, remember Indian green revolution. Indian green revolution, because definitely during the 1960 to 1980, these are the era, these are the what we say, these are the few years, these are the very much important in India, because Mexican variety it is came in India. Now there is a development of this wheat variety, hybrid wheat variety, right? As well as there is a development of some rice wheat variety, and whatever the pioneer work which is related to mutational breeding, it is not possible. If Swaminathan is not there, it is not possible. So whatever the credit is going on for the green revolution in India, so all this credit goes to Dr. M.S. Swaminathan. Okay, and now what happened? First time Dr. Swaminathan used the gamma rays. That's why Dr. M.S. Swaminathan, also called as the father of radiation genetics. Father of radiation genetics. Because first time scientists used the gamma rays, purposefully induce the mutation for the crop improvement. Okay. So definitely see, rays hai, definitely these are the harmful. But now what happened? Purposefully we have to create some changes for the crop improvement. Same, all this parameter, all this study done by the Dr. Swaminathan. So that's why we can say is the father of the plant mutational breeding, means purposefully induce the mutation for crop improvement. For the crop improvement, that's why it is called as the mutational breeding. Use of induced mutation for the crop improvement. So this technique simply it is called as the mutational breeding. And yes, it is required. ऐसे नहीं कि हर बार बेनिफिट ही होगा. Sometimes scientists result get fail. मतलब जो अच्छा था उससे भी worst हो गया. Sometimes what happen, there is a good quality of product, it is get lost. So failure and the success, these are the simultaneous process during this such type of this program. But definitely after the few efforts, we obtain a good result, hybrid variety who is the high yielding and showing the high production productivity is more. It is possible by using the technique that is mutational breeding, and mostly in case of this plant gamma rays is used. Got it? Yes or no? So to explain this experiment, we are taking simple example with respect to wheat variety, that is Lambda Rosa 64A. Should I continue? Okay. So हम एक स्वामीनाथन का एक एक्सपेरिमेंट देख रहे हैं. चपाती, सपोज अगर चपाती रेड कलर की होगी तो कोई खाएगा? रेड कलर चपाती. टेल मी, yes or no? If red color chapati is there, so what is your reaction? Same condition happening before few years, Indian people rejected the red chapati. Why? So we are talking about some experiment of Dr. Swaminathan. Now what happened? We are taking the two varieties, two wheat varieties. Number one, we are taking the one variety, that is Lerma Rosa 64A, और एक variety थी, that is Sonara 64A. Now what happened? Both these are the wheat variety. Sonara 64 also wheat variety. Lerma Rosa 64, these are the wheat variety. दोनों के दोनों wheat के varieties है. Now they have some unique property. Lemna Rosa, basically these are the color of what we say, that is both these are the species, these are the having the red grains. Both they has the red grain color. Normally if you see the wheat color, wheat means if you see the wheat seeds, तो उसका color कुछ अलग सा होता है, means something is wheat color. So that is a wheat grain. But now initially what happened, these are the red color grains are there. But now what happened, one another good quality, they are showing the high amount of yield. High amount of yield. We are talking about, these are the common point for the both the species. Lemna Rosa as well as the Sonara. Okay, because both these are the variety of the wheat and they are having the red color grains, but as well as that is good quality, they having the high amount of this yield. So these are the some specific condition with respect to both these species. But now what happened? Due to red color of that bread or chapatis, Indian people rejected such variety. So we are talking about जब कभी भी इसका हुआ condition is what happened, they produce the red color chapati, they produce the red color bread, red color chapatis, and due to what happened, this red color bread, because of that red color, such crop it is rejected by the Indian peoples. Sorry, automation हो रहा है screen में. Rejected by Indian peoples. Definitely, लाल चपाती कौन खाएगा? मन ही नहीं होगा खाने का. But now what happened? Scientist Swaminathan purposefully create a mutation in both the species. So we are talking about what is the work. So we are taking the two species together. Number one, that is Lerma rosa. रोजा भी दिया हुआ है, रोजो भी दिया हुआ है, रोजो ऑल्सो देयर, दोनों का मीनिंग सेम ही है. लेमना रोजा and the another species that is Sonara 64. So inside these two species, individually, Dr. Swaminathan bombarding the gamma rays mutation. And now what happened? Due to mutation, gamma rays mutation. So now what happened? Due to mutation, whatever we are talking about, that is Lemna rosa 64A, now it is converted into Pusa lerma. इसका नाम चेंज हो गया. After the mutation, इसका नाम हो गया Pusa. Pusa, these are the name of the institute where such type of this mutation process is carried out. So it is converted into Pusa lerma, and that Sonara 64, now it is converted into new name, that is Sharbati Sonara. Species same है, but now what happened? What is the main problem? Red color. Red color सबसे ज्यादा problem था, बाकी कुछ problem नहीं था. Everything is good, amount is high, its amount, protein amount is okay. But now what happened? Due to red color, now Indian people rejected that. So solution क्या दिया यहाँ पे? Solution with respect to create a gamma mutation, and after the gamma mutation there is a development of these two species, Lemna rosa 64A, which is the red color high yielding trait or variety of the wheat. It is now known as the Pusa lerma, and Sonara 64, after the mutation usko naam diya, that is Sharbati Sonara, because it is converted into different species. But now what happened? Everything is same. Now with this two property, एक change हुआ, change with respect to color of the grain. So now they obtain the color that is amber color or the yellow color. So it has both, both this has yellow or amber colored grain. That's it. And now we can easily consume such type of this Pusa lerma and the Sharbati. Sharbati आपने नाम भी सुना होगा. It's a good quality wheat. शरबती मोटे मोटे दाने रहते, ग्रेन इज वेरी लार्ज एंड गुड क्वालिटी, सॉफ्ट रहती है बहुत चपाती. सो इट्स डिपेंड अपॉन, बिकॉज सी मार्केट में जाओगे तो आपको बहुत सारे वैरायटीज के मिलेंगे, बट शरबती दिस आर द गुड क्वालिटी ऑफ व्हीट, मोस्टली इट इज यूज्ड फॉर द व्हीट प्रिपरेशन. या सो दिस आर द वन एक्सपेरिमेंट ऑफ द स्वामीनाथन. इंड्यूस म्यूटेशन किया है बाय यूजिंग द गामा रेज. आई एम रिपीटिंग दिस पॉइंट अगेन बिकॉज ऑलरेडी आस्क्ड वन क्वेश्चन फ्रॉम दिस पॉइंट. लेमना रोजो, रोजो है या फिर रोजा है, depend, okay? Rosa also, some book it is given as a rosa, some book it is given as a rozo, both these are the correct. Lemna rozo or rosa 64A as well as the Sonara 64A, these are the wheat variety having the high yield but red color grains. Now what happened? Red color के grains हो गए, मतलब चपाती आटा जो भी बनेगा red color का बनेगा. So due to that red color, Indian peoples rejected that. Purposefully, Dr. Swaminathan created a gamma mutation, induced the gamma mutation, And after the mutation, what happened? Whatever that red color is there, that red color it is converted into yellow or the umber color. And now that Lemna rosa 64A, after mutation converted into new species, name of the species that is Pusa. पूसा इट इज द नेम ऑफ द इंस्टीट्यूट, दिल्ली में होता है, पूसा रोड भी है वहां पे. पूसा इज द नेम ऑफ द इंस्टीट्यूट वेयर सच टाइप ऑफ दिस म्यूटेशनल ब्रीडिंग एक्सपेरिमेंट इज कैरिड आउट. पूसा नेम ऑफ द इंस्टीट्यूट, सो वहां पे बना, इसलिए इसका नाम दिया, दैट इज पूसा लेरमा. एंड सोनारा 64, दिस नेम गेट चेंज एंड नाउ इट इज कॉल्ड एज द शरबती सोनारा. गॉट इट? Yes, this point is clear everyone. Okay, chalo. So now we are making one chart with respect to induced mutation. We are taking one crop, and then we are talking about varieties, name of the varieties, and how they are showing the resistance activity. Chalo. So next part we are talking about with respect to chart, mutational breeding chart. Divide your page into 3 sections. एक minute. Okay. First column should be small. Second and the third quite larger. Here we are talking about crop name. Now here we are talking about variety name. And finally we are talking about their activity. Means resistance to what? Simply we just discuss. यहाँ पे हमने discuss किया था. There is we are taking the one crop that is wheat. In case of this wheat, Lemna Rosso 64A and the Sonara 64, it is converted into Pusa Lerma and the Sharbati Sonara. And what is the property? Color of the grain, it's get changed. So that is a first wheat variety. Now number second, we are talking about crop. Second crop, that is rice. If you want, you can write down here. First crop, that is wheat. Variety name, that is Pusa Lerma, and the another one, that is Sharbati Sonara. And now what is the activity? That is color of grain changed. Red to umber color. That's it. With a first crop, that is wheat. By using the gamma rays, you have to mention. We are talking about mutational breeding example. Mutational breeding example by using gamma rays. Now, next part, after the wheat, second, what will the staple food in India? That is rice. In case of this rice also, we produce the different species. Number one, we are talking about by using the gamma rays. These are the production of the one species, that is Atomita. Atomita variety. And this Atomita variety, basically these are the resistance to bacterial blight as well as the resistance to brown leaf hopper. So we are talking about what is the property they produce. These are the resistance to, first point, bacterial blight, and the another one, resistance to brown leaf hopper. No question. still asked on the rice variety, so don't worry, it's for your school examination. Another one species that is Jagannath, राइस की स्पीसीज है Jagannath variety, Jagannath species. This Jagannath basically they have some unique property. These are the having resistance activity for the insect. So we are talking about these are the resistance to insect. And the another property they having lodging resistance. Lodging means what? If there is a stem is very delicate and the heighted, so that time what happened? They get bent, easily bent hoga because rice is a monocot. If stem is delicate, so is this any high wind flow or heavy rain? So ultimately what happened? That stem it is showing the bending. So if it is get bent, ultimately there is a loss of productivity. So now what happened? These are the showing resistance to lodging. No bending of the stem due to heavy rain or due to heavy wind. So these are the second species of the rice, that is Jagannath. And the last one we are talking about Remi. Last one, Remi. Remi basically these are the showing resistance activity to early maturing. It's about the early maturation. Resistance to early maturation, and as well as the number of the inflorescence, these are the more. Because see, if inflorescence more, it means that productivity also more. So number of inflorescence so number of inflorescence is more. So these are the three rice variety. And abhi tak jo question bana hai, that question is coming from the wheat, and the second question it is coming from the moong. So we are not taking too much detail about the rice, just it's for your school examination. Now third crop which is produced by the mutational breeding by using the gamma rays, that is cotton. Third one, third crop, that is cotton. Cotton basically the name of this variety, that is Indore 2. Indore 2. And now this Indore 2 basically it will be showing resistance to cotton ball worm. Resistance to cotton ball worm. As well as we are taking the one species with respect to castor. Next crop, castor. Name of this variety, that is Aruna. And this Aruna it is having more oil content. More oil content. Fifth crop, groundnut. Crop, that is groundnut. Species no, not mentioned, so simply dash. And in case of this groundnut, basically there is a production of the thick shell of groundnut. So definitely if groundnut shell is thick, so ultimately the seed is get protected from the various pathogens. So shell. Now the next one, number 6, peppermint, that is mentha. Next one, peppermint. Again, dash in variety name. Basically, this peppermint basically is having one property. It is having the high oil content, and as well as these are showing resistance to disease. And the last one we are talking about, जहाँ से question बना था in 2015, that is मूंग. अभी तक दो ही क्वेश्चन बने यहाँ से, one is wheat and another one is मूंग. So we are not taking chance. अगर दिया है तो हमें सब कुछ स्टडी करना पड़ेगा. So in case of this मूंग, basically we are talking about these are the one specific type of this species. Those are showing resistance activity to yellow mosaic variety. Variety name, just give the dash. Name is not mentioned. But now we are talking about these are showing resistance to virus. So I'm showing first point resistance to YMV, that is yellow mosaic virus. And this yellow mosaic virus sometime option में इसको इस तरह से देंगे YMV. YMV, these are the specific type of the viruses who infect the moong plant, means other plant also. But now with the help of that gamma mutation, gamma rays exposure and the mutation, creation of the mutation, such type of this moong species, these are the resistance to viruses. Name of the virus, that is yellow mosaic virus. जैसे tobacco mosaic virus होता है, उसी तरह से yellow mosaic virus है. Now, as well as it will be showing resistance to powdery mildew. Powdery mildew. And this powdery mildew, nothing but we are talking about, these are the one fungal disease. Fungal disease. So it will be showing resistance activity to fungus, moong plant showing resistance activity to fungus, as well as it will be showing resistance activity to fungi also. Sorry, virus also. That is yellow mosaic virus. So अब तक का हमने pattern देखा. So there is only two question is arise. One very important, that is moong. Another one, that is wheat variety. So from only two crops last year question बने थे, one question, not more than that. Middle wale, this was for your school examination. For the need, no need to preparation. Okay, Kinmin, this point is clear everyone. I know these are the quite lengthy, पर आपको करना पड़ेगा. Best way, solve the previous year questions. आपको एक idea आ जाएगा कि कहाँ से कितना point करना है. What is the limitation? Because plant breeding, hybridization, it is very wide branch. क्या क्या करोगे? So do whatever question is coming from that point. That's it. Only those part, not all. Okay, chalo. So we cover the hybridization. We cover the what we say, polyploidy. Then we cover the mutational breeding. एक point बचता है selection. Selection point I'll continue the next class, means next week. And whatever the portion, थोड़ा सा ही part बचा हुआ है, because now we are taking some chart. Fungal resistance plant, disease resistance plant, virus resistance plant, eco-friendly plant, means they showing the resistance activity, but they never kill the insect. Eco-friendly plants. So we are taking some chart wise information, and that chart is very important. Okay, chalo. So is there any doubt from today's class? Any doubt? Okay, chalo. So please revise this part, again and again. And telling you only one, maximum two questions are there. Minor chapter है, ज़्यादा इस chapter का stress नहीं लेना है. थोड़ा सा lengthy है, but solve the previous year questions, last 10 years question, and try to catch what is the exactly important area. हमने आपको बोला है, but now what happen if you do yourself, तो आपको clear cut idea बन जाएगा, right? Because question in front of you, theory is there, and now below the questions options are there, so you can correlate with each other. Because बहुत बार यहाँ पे होने वाला है पूसा 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 सदाबहार, पूसा पूसा शरबती, पूसा लेरमा, बहुत बार पूसा पूसा आएगा. So this chapter is again required the revision to understand the name. Okay, चलो ठीक है. Now yeah, so with this we end this session. Rest portion, 6th class. Yeah, thank you so much.